Hello people. So the intestinal mucosal barrier or the intestinal barrier is a specific part of the GI associated lymphoid tissue that plays a significant part in the defense of our gut and our body of different microorganisms present in our GI tract. First, before we start, I need to mention some terms. First, it is this term called MALT. MALT basically means the mucosa associated lymphoid tissue, and it represents that we have in our mucosa different lymphatic tissue, lymphoid tissue that is shielding our body from pathogens that are present on the borders of our body. So this is a superior term because it can be further subdivided. It can be subdivided into organized and unorganized. Most of the MALT tissue is unorganized, so different cells in the mucosa and different mechanisms of defense. There are also organized parts, for example, Peyer's patches in the intestines, but it can also be subdivided by location. So, for example, in our case, we have GALT, so gut-associated lymphoid tissue, and the point I'm making, MALT is a superior term, so mucosa associated lymphoid tissue is having these subdivisions like GALT or the gut associated lymphoid tissue, or like BALT, which means bronchus associated lymphoid tissue. And even the skin, SALT, is part of MALT. And there are many others. So the intestinal barrier is actually a part of the GALT tissue. We will now see what are the important elements, the important cells, their products and processes that are happening in the mucosa that are shielding us from all these pathogens present in the lumen. In the picture, we can appreciate then that up here we have, this will represent the lumen. So this is the lumen of our intestines. Then we have on the apical surfaces of the cells, we have the mucus layer. So this would be the mucus layer. In the colon, this layer has two parts, an outer part and a sub part or inner part. In the small intestine, this is only one continuous part. Then we have the cellular layer. The cellular layer is simple columnar epithelium. And then this last layer is actually representing the lamina propria of the mucosis. So the lamina propria of the mucosis. And as we can see, the lamina propria has different cells, many capillaries, the extracellular matrix and lymphatic capillaries, and also other properties. So now we will start dissect this intestinal barrier and what it is made of. So the first element I want to talk about are actually the cells. So the cells form this physical barrier of the simple columnar epithelium. And in between every of these cells are special molecules called junctional complexes. They form tight connections that don't allow paracellular transport or diffusion, so the cells can selectively take up molecules from the lumen and don't let other things go through this paracellular gap. For these connections that are actually present between these cells are called tight junctions. So tight junctions are one possibility. Then we also have adherent junctions, and we have desmosomes. So these three types of connections are enabling these cells to have this continuous row with these physical properties that are compact and that regulate that these cells stay together and that they don't let anything pass beside them. The second important thing are the cells themselves. So these cells have special properties, and the first cell I want to talk about is the goblet cell. So this is a goblet cell. It is shaped like a goblet or a cup, and that's why it's called the goblet cell. So this is a goblet cell, and we see that the cell is polarized. So it means on one pole the nucleus and most of the organelles are sitting, and in this cell this is the basal pole. This would be the basal membrane, and this is the basal pole, and here are the organelles. The apical part has many of these granules, and these granules continuously release their product, and their product are mucin proteins, so mucins. These mucin proteins are glycoproteins, so mucin. These glycoproteins have many negatively charged parts, and they also have the possibility to bind water. And with water, together with water, they are forming the protective mucus. So the mucus is a gel-like substance that covers the apical part of the cells. In the colon, we have the outer part of the mucus. So in the colon, we have the outer part and the inner part. This outer part is loose and it is infiltrated with bacterial microorganisms, and the inner part is compact and sterile. In the small intestine, we have only one layer. The mucus is different in different parts of our body because these cells produce many types of mucin proteins. When it comes to the intestinal barrier, the mucin that is produced contains the mucin protein 2. But for example, in our airways, we have the mucin protein 5. This would be a difference. So we have distinct forms of mucin. Also, this barrier, so the mucus barrier, is actually about 200 micrometers thick. So the goblet cells have this possibility to produce these mucins who bind with water and produce mucus that is definitely shielding these cells and making it hard for the pathogens to come near them. Now, and this mucus is continuously secreted. Now, the second cell I want to talk about is the Paneth cell. So this would be a representation of a Paneth cell. A Paneth cell. So the Paneth cell is also a very interesting and important cell, And it also has the nucleus and the organelles to the basal surface, and the granules are apically. This cell does not release its granules continuously, but it releases them when it comes in contact with bacterial antigens or with bacteria. And then we have the secretion of this content. The Paneth cells are also sitting near stem cells in Liberkin crypt glands, and it is theorized that they also protect this vital population of cells, because stem cells are the cells that renew our intestinal epithelium. Also, these granules contain special antimicrobial proteins and compounds, and they are important in the fight against mostly bacterial pathogens, and they have a possibility of destroying many microorganisms. So one of the proteins is alpha defensin. Another is lysozyme. So alpha defensin is a protein that basically has many positively charged domains, and it can insert itself into the membrane of microorganisms, and the membrane of bacteria has many phospholipids that are negatively charged. And by inserting itself in the membrane, it will disrupt the membrane function and thus probably destroy the bacteria. Lysozyme is actually not, it is a specific enzyme. So this specific enzyme acts on the bacterial peptidoglycan. In order to explain this, I will insert another picture. So this would be a gram-positive bacteria. So this gram-positive bacteria has this cell membrane. So this is the cell membrane of the gram-positive bacteria. And this structure is actually the cell wall of the bacteria. So the bacteria has this protective cell wall in its structure. If we would enlarge the cell wall, we see that it is built out of special molecules, and that it is actually a polymer form of these molecules that make up this mesh-like layer on the outside of the bacterial membrane. So these molecules, this molecule is called NAM, in short for N-acetylmuramic acid. Acetyl acid. This molecule, together with this molecule that is called NAG, or the N-acetylglycosamine. The N-acetyl together, these two molecules have this bond. They fuse together with this bond, so this bond, and this bond is actually a beta 1,4 bond. So the first and the fourth carbon molecule in these molecules make a bond, and our enzyme, so if we come together, if we come back in this picture, we see we have this enzyme that the Paneth cells are secreting, lysozyme. This lysozyme enzyme is acting on this bond. So the lysozyme enzyme comes in here, and it disrupts this band. This would be the lysozyme. So it actually breaks up this connection between these two molecules, and then the peptidoglycan wall destabilizes and breaks, and the fragile cell membrane is then exposed. So this is another way that our intestinal barrier is working through the Paneth cells. The second important cell I want to talk about is the microfold cell. So this would be a microfold cell, this cell. The microfold cells are called like that because they have actually these folds into the membrane, and these cells, microfold cells, are actually special antigen presenting cells. So antigen presenting cells. These cells take up antigens from the lumen, and then they are actually presenting them to, for example, intraepithelial T lymphocytes. So this cell is an intraepithelial T lymphocyte. So these cells are special antigen presenting cells. When they take up the antigen, they present it to this lymphocyte, and then the lymphocyte decides what is the future of the immune response. Also, besides the microfold cells, we have these cells. These cells are actually dendritic cells. The dendritic cell is also a specialized antigen presenting cell, and it can also present to lymphocytes that are in the lamina propria or to other lymphocytes the antigens. These cells can also travel to lymphoid organs like lymph nodes and present there in the gut lymph nodes their antigens. Also, we have the macrophages. The macrophage, so this would be a macrophage. These cells can also act as antigen presenting cells, but they have also the function of phagocytosis. So if for example, a bacteria enters into the lamina propria, maybe the immune response only of the macrophage will be enough to eliminate it. Then there is the next element that is important, is that if the immune response develops on some form of antigen present in the lumen of our intestines, and if it activates the production, it can activate the production of immunoglobulins, protective immunoglobulins that are especially designed to act on mucosal surfaces. So, if, for example, the dendritic cell presents an antigen to this T lymphocyte, and this T lymphocyte can then turn into a Th2 lymphocyte, and it can produce special molecules, cytokines. So the naive T helper cell produces this T helper cell 2, and then it produces transforming growth factor beta. Transforming growth factor beta has this possibility to make B cells switch from IgM production to IgA production. And then also with interleukin 5, B lymphocytes are actually transforming into this cell, and this cell is called a plasma cell. The plasma cells are actually cells that produce and secrete antibodies. And if our immune cells here stimulate this production, these cells start producing special molecules called IgA, or immunoglobulin A molecules. Immunoglobulin A is found in our bloodstream but the main type of immunoglobulin is actually being secreted outside into the mucus. So here we will see how our enterocytes, or the main cell in this epithelium, plays also a role in the intestinal barrier. So if I insert this picture, we can appreciate that first our IgA has been produced. So we have the plasma cell that produces this molecule, or IgA. This IgA is a dimeric molecule, has these two elements, and in order to get into the enterocyte, so this would be an enterocyte cell, in order to get into this cell, these antibodies need to connect to this protein. This protein is called polymeric IgR, or immunoglobulin receptor. So polymeric IgR. When the immunoglobulin A connects with this protein, they internalize into the cell. Endocytosis is happening, and then with transcytosis, so transcytosis, they are being transported from the basal part of the enterocyte to the apical part, and then they are being secreted. But before the secretion, the protein is split, so this would be the secreted IgA, and this part of the protein is left on the IgA. It is called the secretory component. And this molecule is called secretory IgA. This is how IgA actually comes into this mucus layer. IgA is a special molecule. When it is produced, transcytosed through the enterocyte, so this would be an enterocyte. When it is being transcytosed and synthesized here and secreted here into the lumen, this molecule has now the potential to bind to the antigen it was actually secreted for. So, for example, our M cell has found an antigen from this bacteria. It has alerted our immune response to this antigen, and the immune response has delegated to producing these antibodies. Plasma cells have been created. The IgA molecule has been synthesized and released, and then it bound to these enterocytes. It was then pushed with transcytosis into the lumen and into the mucus. Now these antibodies have the possibility to bind to these antigens present on the bacteria. What are they doing with that? These antibodies, or IgA molecules, secretory IgA molecules, have actually not that good of a capacity to bind complement proteins, and also it is hard to find any here. These antibodies have the possibility to block the antigens, they are the receptors, the proteins on these bacteria. Thus, the bacteria cannot communicate with other bacteria, and they can also not bind to our cells. And that is how they are enabling the possibility of destroying the function of these bacteria. So these would be, for example, gram-negative, and these would be gram-positive bacteria that are present in the lumen normally, but also they can be pathogenical microorganisms present in our gut. So this is actually a simple overview of how this functions. Also, the capacity of bringing in more immunological cells is happening through these blood vessels, and there are many in the lamina propria, and the immune cells, when they are attracted through chemokines and cytokines, they are entering the lamina propria. Also, when our cells need to go to the lymph nodes, so this would be a capillary, a vascular capillary, and this would be, for example, a lymphoid capillary. And when our cells need to go to the lymph nodes, they travel to the lymph capillaries. So this is a simple overview of the intestinal barrier. Thank you.